The next concept that we are going to look at is look at the oxidation of fats that occur in the peroxisome rather than in the mitochondria. And so there's a compare-contrast here. Mitochondria, obviously, the carbons are going to be converted into electron carriers so that we can create ATP. And all the carbons, whether it be in the degradative process of the fat itself or all the way through the acetyl-CoA and citric acid cycle, we're going to make ATP with those. In general, though, we do need acetyl-CoA for a couple of places where we do synthesis. And we typically don't take the acetyl-CoA from the mitochondria to do that, but what we can do is we can take the acetyl-CoA from fats if they're degraded in the cytosol, because the acetyl-CoA that is produced in the cytosol has the potential of being part of synthetic processes, including cholesterol. And so we have the process that occurs. Now, remember, as we do the oxidation, the hydration, the oxidation, and then the thiolysis, those 4 steps, we're going to create electron carriers and we're going to create potential energy. But in this particular case, what we're going to do is we're going to allow the electrons that are being produced in the FAD to be accepted by oxygen and then converted into water. And so we tend to lose the potential energy that can be seen here from the process to water rather than to making energy. The NAD that is produced has the ability to be used in other places and can actually transport, like the NADH, from glycolysis into the mitochondria. So we can extract some of the energy from this process. But the ultimate fate of this acetyl-CoA is for synthetic processes. Biosynthesis of precursors in plants. So fatty acid oxidation doesn't occur in the mitochondria of plants because that's not necessary in terms of creating a proton gradient there to create energy. We know we create energy totally differently in plants. But there are some cases in which we do need to break down fats and allow energy to be extracted. And what really is part of this process is when you're in the seed form and you're buried under the ground, before you can reach up and start to capture light energy, the seed and the energy stored as fat is converted through beta oxidation to acetyl-CoA. The acetyl-CoA then goes through the alternative citric acid cycle in the plants to create oxaloacetate, and then the oxaloacetate goes into gluconeogenesis. And then from that, you have both the structural components, the sugars, to actually grow stems and stalks and go above ground. You're also going to be able to derive the amino acids, energy, and all the metabolites that you need from that glucose. And so we're going to get fats to glucose in plants where that is not the case in mammalian systems. This alternative citric acid cycle is what allows us, or allows plants to be able to take fat and make sugar where we don't have that pathway. There is an alternative oxidation scheme that can happen in the endoplasmic reticulum of the liver and kidney. And what actually occurs is a oxidation at the omega carbon. And you see the reaction with oxygen to make hydroxide. We oxidize that up to an aldehyde using another equivalent of NADH. We create, and water, we create the acid. And so beta oxidation can occur from both ends of the fat, and we end up with intermediates, one of which is a citric cycle intermediate. And so oddball. Just wanted to let you know that that has been seen and is in the body, but only in certain tissue types. Now, here is something that is challenging, and for quite a long time we thought it was just a disease state. We now know that the formation of what are called ketone bodies are important. They are an energy source, an exclusive energy source for certain tissue types, and ketone bodies were first discovered really because of this. A patient comes in, and as they breathe out or as they're sweating, what you can smell is a fruity smell. Most people would describe it that way if they haven't been in an organic lab, but if you've been in an organic lab, what you smell is acetone. And so what's happening is in the biochemical process, the body will produce acetone in the bloodstream. Now, do we suggest you go into the organic lab, grab the red wash bottle and suck on the tube there? No, acetone is not a molecule you want in the bloodstream. It's relatively toxic. But there's a byproduct of a synthetic pathway that if you allow it to go too far, you will start to have a breakdown, non-enzymatic, into acetone as one of the byproducts. When you actually smell that, what you're usually attributing this to is a unregulated diabetic, okay? And what the mechanism happens is the insulin is not being signaled right, the sugars may not be processed right or brought into the cell. The cell feels like it's under starvation. And so if it feels like it has no sugar, then what will happen is you start mobilizing fats. But the problem with that is that you're sending mixed signals to the cell, and you start mobilizing lots and lots of fat. And whether or not you are starving yourself or you're under a strain because of unregulated diabetes, this is what happens, okay? The cell tends to think, because the concentration of glucose is low in the cell, it starts to take off all the oxaloacetate out of the citric acid cycle and go up to gluconeogenesis to start sending glucose out, okay? And so either starvation state, long-term starvation state, or the misprocessing of the insulin starts sending out glucose, okay? The problem is I still need energy, and so the signaling gets mixed up, and I'm mobilizing fat stores, and I'm bringing in fat inside the cytosol. The fat goes into the mitochondria, and the fat gets broken down through beta oxidation. I get lots of acetyl-CoAs. But this is what I have, a lot of acetyl-CoA, and I have, at this point in time, none of this, okay? One thing they don't show you here is that you're also mobilizing amino acids. At this point in time, amino acids are going to feed here into the citric acid cycle, but they'll go out as oxaloacetate. All right? So quite often, when you shift your metabolism from using quick glucose stores, whether it's glucose in the blood or glycogen, the shift is not just to fat, but you also start using protein. And in a well-conditioned person, you actually start using protein and fat at about 50-50%, depending upon if you are very well, if your metabolism is working right. And that's connected to your conditioning level. All right. What happens now is the acetyl-CoA is built up in a high concentration. And it never stops because the cell still seems like it's being starved. What happens is, if acetyl-CoA cannot react with oxaloacetate, it starts reacting with itself. And so we create a class of molecules called the ketone bodies, which then will be sent out to the bloodstream In times of starvation, the muscle can use that as energy, but there is a small amount of ketone bodies that are always synthesized at a very low level, and the heart and the kidney actually use that more specifically for energy than it will use glucose. And so if you think about the function of you in being alive, your brain has to work, glucose. Your heart has to keep pumping, ketone bodies. And so we can extract the essential energies from two different sources to keep the most vital two things going. And so for a long time, we didn't know the heart used that exclusively or preferentially, and we didn't know these molecules were important. We only found it when it got too much in the bloodstream and acetone was being produced, because one of these products in the ketone bodies will degrade into acetone and another product. I'll show you that just in a moment. This particular process and this biochemistry is in the liver cell, and this occurs in liver. So the ketone bodies, I told you we would have ketones, or the acetyl-CoAs reacting together. The ketothialysis is the enzyme, and so before we use the thialysis or thioase here, where we had the sulfur attacking, what's going to happen here is a sulfur is going to be a leaving group. And so if I do some inventory, and that's what you need to do, okay, like if I put this on the exam, this particular slide, and I say, write the mechanism, okay, what is the first thing that you do other than panic, cry, and then finally decide, I probably should put something on paper, okay? So you've gone through all of those emotions. What do you do? What is the best thing to do? Take an inventory. And so I'm going to take an inventory. I'm going to call this blue, and I would translate that one as this one. And I'm going to go, this is blue, that's blue, okay. And then what I see if I go on the other, this is green, that's green, this is green, that's green, ketone is green, ketone's green, and there and there. Okay, I see a change in one place that is fundamental, okay, in terms of the green, and I see a change which is fundamental in the blue. All right, in the green, this in the product is different than this. What is the difference between this carbon in the reactant and this carbon in the product? The amounts of hydrogens. What's the difference between this carbon ketone and this carbon ketone? It does have an alpha carbon, but it's not connected to the sulfur. It's now connected to a carbon. So this must leave, and this blue ketone is now connected to the green, okay? And so what you predict is I'm going to have to deprotonate this carbon, all right? Is there any organic reason why deprotonation at this carbon is stable? It is what to a carbonyl? Alpha to a carbonyl. So there is a reasonable idea of, okay, if that does get deprotonated, the structure stabilizes it. And so every time you have a mechanism, you can have an acid and you can have a base, all right? And so if I write that, okay, base, all right, and acid. I'm going to put this on the slide in no particular place, okay? All righty. Ready? Okay. Everybody ready? All right, let's try something new. Why don't you show me what this does? Go up to the iPad and draw the mechanism. we're all going to try a little bit of this. Now, when you're given the pencil to go to the iPad, you have two jobs. One is to draw the mechanism. That's a simple one. The other one is social. There's some social pressure with this. After you've drawn your mechanism, you have to give the pencil to somebody else because they get to do it. Okay? So what would you do to draw this mechanism? I like best guesses. Okay, deprotonate. Yes, attack the carbonyl. Beautiful. Up, down, and that goes into the sulfur. And there we go. Picks up the proton. And you have perfectly wrote the mechanism. You've done one of your two jobs. Okay? Go select another volunteer. Okay? That was great. All right. Now, in this mechanism, I would have accepted, okay, if you see the arrow onto the sulfur and then the sulfur picks up a proton, I would have also accepted the bond between the carbonyl and the sulfur to directly go and pick up that proton. And so either one of those is absolutely acceptable. I don't care if you show me more arrows than less arrows, but that articulated that movement about what would happen. The reason why I don't write it on the sulfur too often is because in the reaction, the proton is so optimized to pick that up that it will directly go to it. But in an organic mechanism, what you would do is you would put that negative charge on that sulfur for a little while. It would roam around until it found a proton. Yes?
Also, technically, couldn't use the sulfur to deprotonate it because, like, the H could be grabbed up by something in the system, like what it happens in the last step there.
And so pick up which sulfur?
The sulfur that they have, like, above the arrows. Couldn't you technically use that as, like, a base?
Yes, but it really doesn't happen that way in the enzyme. In an organic flash, yes, you would have the two interacting. But remember, we're in the enzyme that is catalytic. So one of the amino acids is going to act as the base, and one amino acid side chain is going to act as the acid. And so if you're writing an organic mechanism between these two entities, more likely it is going to be that way because you have to have it in a, in this case, you could do it through an acidic mechanism or a basic mechanism, and so in one of those cases you may have to use that as the base. But inside the enzyme, the enzyme is going to act as that. All right? Okay. The next step is, remember, we've got lots of acetyl-CoAs, and so that molecule that you just reacted is going to react again with another acetyl-CoA. And so, let me take this for just a second, all right? And so let's take a look and do some inventory, and I'm going to, that looks no different here, no different here, and uh-oh, that looks weird, that's different, and then that one is that, okay? And then I go, and if I look at the other, the CoA, okay, there's the CoA, here's the sulfur, there's the ketone, there's the ketone, and there's a red, and there's a red. All right, there are two possibilities in which you can attack the acetyl-CoA, because I have a ketone here and a ketone there. And we've now identified it's going to be this one that is different than that one. Okay. And then we have the red methyl here is now red with 2 protons. And so I'm going to, in no particular place, put a base, and absolutely in no particular place, put a proton on the slide. All right. Are you ready? I know. It feels so weird when you have to do it. All right.
Okay. It's not going to be like this.
Okay.
Just going to work.
Good.
You said this one was the.
Mm-hmm. Correct. And where does it attack? It likes to attack at things that are partially positive.
That one.
Is it there?
Ooh, I don't know. Is it right there?
I believe it's the other. So hit the back arrow there. And back one more time. And go ahead. Because a negative charge loves to attack things that are partially positive. That's the carbon of the ketone. Now look at the product. When you attack that carbon to that, what was a carbonyl, a lot of times we kick up and kick down, right?
So you kick up and then kick out.
But if you kick up and kick down, I would have a ketone back, but I don't have a ketone. So what's the other thing that you can do? You can kick up and then pick up a proton.
Ooh, this is hard to control.
There you go. And that's the first half of your job. Okay, and we create the intermediate. Hydroxymethylglutaryl-CoA synthetase. Ooh, lovely. Oh, no, you're touching it now? Oh, wait. Did you intend? Okay, all right. There was intent behind that. All right. All right. As we look at this, come on up. Be with me. All right. What we're going to do is lose a CoA, right? And so I'm going to identify where the changes are. So all the way up through here and including this ketone is still in the product. It's right here where I have a sulfur and a CoA. What do I get over here? An anion, an oxygen. Okay. And the reactant is water in this, and the leaving group will be CoA. All right, so I am in no particular place going to place a base and an acid. That's probably not a good place to put it. In no particular place, but that's not a good place for no particular. I'm just going to randomly put it right. Well, if I, stop. It went too far. No particular place is probably better there.
Could I attack the water?
You can attack the water.
Here.
Yes. Now I have a hydroxide
Yes.
that can be a nucleophile.
Hydroxide will then attack here.
Okay. Yes.
Kick off. Or, well,
Yeah.
that's kicking off.
Mm-hmm.
Which was,
Oh,
okay. Whoops.
come on.
Okay.
Oh,
Bad showing. But do I need to draw it again?
nope.
Okay.
Just,
Well, the acetyl-CoA kicks off.
uh-huh.
And
And the
then
sulfur is negatively charged, but we want a product that has,
So the OH, well, no, there's already an OH attached to it.
so the sulfur is negatively charged. Up here in the leaving group, it is CoA sulfur plus a hydrogen. So that negative is going to get with a positive
Oh, I didn't even see that. Okay. So in this,
in no particular place. Yes.
there.
Perfect. You've done it.
But
Mm-hmm.
the oxygen that's attacked,
Yes.
but then doesn't have to.
Okay, so yeah, here's the question that you have. He attacked a hydroxide, right? And so after his electron movement and what he showed, the structure that should have been the product, he drew, oops, sorry. Yeah, if you touch it, it moves. He drew that, okay, hydroxide. But what is that structural motif? It's a what? What is that at the end there, the functional group? Carboxylic acid. The pKa's of carboxylic acids are in the 4 range, As soon as it's released from the enzyme, it's going to be in a pH of a little slightly over 7, and that will be much more than a full unit away or difference, and it will become deprotonated. And so acids in solution, if you actually create one, will deprotonate, not mechanistically, but because of the difference between the pKa value and the pH of the solution, the buffering system. And so that's why you will see quite often things that might have been a hydroxide become negatively charged, and the protonation state is actually kind of important. Yes, originally a hydroxide would attack and be there, but it will deprotonate very quickly. Thank you. Oh, nope, you got a job. Yes. Oh, yes. Once you've touched it in a lecture, you don't get to touch it until everybody's touched it. Okay? All right. Here we go. What in the world is happening here? Okay, let me do something for you. All right, I'm randomly going to place a base and a proton in no particular order on the slide. Okay? Beautiful. And that pair of electrons, okay, if you take a look at the reactant, it's a hydroxide. What is it in the product? It becomes a what? A ketone. So take that pair of electrons, make that ketone. Uh-oh, you now have drawn 5 bonds to carbon. What has to happen? It has to leave. And which pair of electrons have to leave? Yes, that's correct. You've completed the mechanism absolutely perfectly. That's half your job. Go find somebody. Oh, yeah, now, this is the hard part, giving it to the next person. If you give it to me, you cannot refuse it. You cannot refuse it. Don't. Okay, wait. I'm not exactly sure we have a mechanism next, okay? But hold on for just a second. Yeah, I'll get it if I need it, okay? We've got acetoacetate, okay, in this particular reaction. And the hydroxymethylglutaryl-CoA cleavage enzyme is what does this, okay? And the acetoacetate is effectively 2 acetyl-CoAs that are floating around, and there's potential energy there, okay? Now, I do have 2 possibilities for a reaction in the next step. Okay, come on up now, we do. But you have a choice. There are 2 mechanisms, and you get to choose whichever one you want to try to go down. All right? The top one is done by an enzyme. This is the one that we really want to have happen, okay? But the non-enzymatic pathway, if you get a lot of acetoacetate in the bloodstream, that overwhelms the dehydrogenase. What can happen if it sits around too long? It will decarboxylate into acetone and carbon dioxide. And this is where the disease state comes in. If you're creating acetone in your body, that is not good. So, do you want to do the top one or do you want to do the bottom one? Okay, the top one. All right. The top one is a dehydrogenase. So that means we have a hydride moving. And in this case, you see we start off with an NADH and we go to an NAD+. So that is going to donate a hydride in the mechanism. And there's a proton. So the reaction, when you see the NADH going to NAD+, is a hydride is going to attack. Now we've got to decide where it's going to attack. It is a good question. This is like a good test question.
Oh, no.
Oh, yes. Okay, so,
In that panic mode?
yeah, you're in that. You really are in that panic mode. So, I'm really preparing you for an exam question. So, what do you do when you are in that panic mode? Little tear comes down.
Looked at you during the exam.
What are we supposed to do? Compare what? Yeah, inventory it.
So the bottom looks the same.
Yeah, the bottom looks the same.
And that one's the same.
Yep. So what's different?
The nitrogen comes off.
There's a hydrogen there. Okay, there's a hydrogen on the carbon, and I'm going to show this so everybody sees what we're talking about. She identified there's a hydrogen here, right? How many hydrogens are on that carbon? None. Okay, but here in the product there's one, right? Also, the double bond is here, but it's gone there, and the oxygen has nothing bound, but now has a hydrogen bound. So this is the only place that changes, and we find that an extra hydrogen is there, and I have a hydride that is negative that likes to attack things or becomes a nucleophile. And
So
but,
oh, sorry.
yeah, go ahead. Yes, you can.
Sorry, I'm shedding.
You're shedding.
So can I
You're
do
getting
A?
really nervous now. Come on. Okay.
Okay.
And so,
Yes.
most of the time we have to have a base. But when you took inventory, do you have less protons in the product? No, we have more protons. So that means that NADH is going to take that bond between the D and the H, and it is going to be a hydride that attacks.
Can you say that one more time?
The electron pair between the D and the H and NADH becomes the attacking pair of electrons. And it's going to attack at a place that is slightly positive. Yes. Okay, remember when we attack a carbonyl, we could kick up and kick down. That's a possibility. Or we could kick up and pick up a proton. In this case, what do we do? Yes, because we have a hydroxide at the end.
So is that a possibility to get that?
Yes.
I'm gonna use a different color.
You crossed the line.
Oh, no,
But that's okay.
that's right.
That's fine. Oh, you've really done it now.
Hold on. The hands are shaking.
Yes. As well on the test.
Okay, watch this.
Watch this. I'm watching.
Ooh, I didn't touch anything.
Oh, there it is. That is beautiful. It is done completely correctly.
Oh, it's done?
And you're done.
Thank the Lord. Can I do my other job?
Yeah, you get to do your other job now. Woo! Thank you. You've now left the next person, no choice. Okay? I feel the joy and the love that is happening. Okay? I'll take this. All right, in the next mechanism, we're going to interact with the other half of the molecule. All right? And I'm going to go ahead and place a proton in no particular place. And so, in terms of your mechanism, all right, you could attack with what? Hydrogens never attack. It's always electrons that are moving around. So then we have to push the electrons. Okay, let me do something else for you. Okay, going back to GChem 1, if we write good Lewis structures, which I know you can, all of you can write a Lewis structure, okay? 1, 2, 1, 2. That's an oxygen that is neutral. An oxygen that has a negative charge has 3 pairs of electrons around it, and that's why the negative charge happens. And so, if you take a look at oxygen, carbon, oxygen, oxygen, carbon, oxygen. All right, what would you do with a pair of electrons? Remember, we have to start with electrons. Let's make one of those pair of electrons on that oxygen has to do something. Make that double bond. Yes. And, well, not at the carbonyl, because the electron pair, it has to be between, so put it right, tack it right to the line to show that it's going to become a double bond. All right, that is great. Now, the carbon of the carbonyl now has 5 things bound to it, so something has to leave. A pair of electrons has to leave. Okay, so in the product, I'll take this for a second. In the product, what you have is 2 pairs of electrons, and you've taken a pair to make a double bond. 2 pairs of electrons and a pair to make a double bond. A carbon, a double bond, and 2 pairs. So that has to stay, these have to stay. So, and these have to stay, and that has to stay. So the only one that is gone is this one. So you're going to do something with that pair of electrons. Mm-hmm. And, okay, let's do just a little bit more inventory. Okay. Here's a carbon with 3 hydrogens. How many hydrogens are there? Not 2. 3. Mm-hmm. Here's a carbonyl that has no hydrogens. Carbonyl, no hydrogens. How many hydrogens are right there? 3. How many are right there right as we draw it? 2. So it needs 1 more hydrogen. Yes, randomly, just magically on the page, no particular place. Wait a minute. You just took, it would be hard to interpret this. Which pair of electrons took that proton? Because you wrote it at the carbon, is it that pair that took it, or is it this pair that took it? Oh, perfect. Show that. Okay, show exactly what you showed me. Okay, go ahead. Yes, that pair of electrons that was bound to the carbonyl now picks up the proton, and absolutely perfect. All right, go ahead and take that and choose someone else. No, she's already touched it this time, so she doesn't get to touch it again until everybody else does. Wait, what? Oh no, I have it. No mechanism. Okay, let me grab it. But we know next time you're on deck. You're on deck. All right. So now that we've gone through a couple of mechanisms, the top end, what we really need is the 3-hydroxybutyrate and the acetoacetate. Those can be transported out in the bloodstream and used as energy molecules. The acetone itself is not optimized, okay? And you only get that if you have synthesized a lot of ketone bodies, much more than what the body needs. For a very long time, we believed that the ketone bodies were a byproduct of a diseased state, but we actually now know that heart muscle and the renal cortex are the ones that use it in preference to glucose. Although the heart and the renal cortex can use glucose, it really prefers the other and will only adapt to use glucose in very strange circumstances. The brain will start utilizing acetoacetate, but only under very long periods of starvation. And we're talking now past 2 weeks into the 3rd and 4th week, where the brain will start using that, because in the bloodstream, the energy molecules are not glucose anymore. The energy molecules are all these ketone bodies.
So like for an uncontrolled diabetic,
Yep.
like technically there are some benefits because you'll get like stronger heart muscle that can use like different products, or you just be like tired all the time? Is there any sort of benefit?
Okay, there's an
Or like
unintended.
doing like a keto diet or something like
Okay,
that.
yeah.
'Cause, like,
All right, for a diabetic, what's going to happen is they will lose a lot of weight very quickly, because what you're doing is, as long you're taking in calories, but you're not extracting anything from it. You're excreting all that sugar, and what you're doing is using your fat stores. Quite often, the increased urination, because you're bringing out all that glucose molecules with all that water, and quick weight loss is an indicator that you've gone into a state of diabetes. So it's a good indicator when people have gone over the threshold. It doesn't, I mean, losing weight is always a good thing, but in this mechanism, it really isn't. Not only does the ketone bodies produce energy molecules, it also acidifies the bloodstream. So that starts to mess things up too. And so ketone bodies in the bloodstream at high concentrations are not good. Very low concentrations of ketone bodies are absorbed only in a few tissue types. So I don't really need to have a large concentration of them. And so I'll uptake those and take them out by those 2 tissue types. You only get into this state. It's kind of like a state in which the car's not running well, but at least you're running. And so when you get to that point where you are in long periods of starvation, then other tissue types start taking it up. It's less efficient. You're not getting as much energy. You would feel tired, and you start becoming lethargic. You're also changing the entire biochemistry. Next semester, we do talk about the keto diet, though.
Oh, okay.
Yeah, because what you're doing in the keto diet is simulating a starvation condition. And you're forcing the body to start taking in fats that then have to be burned up. And so it tends to take proteins from other places to feed the citric acid cycle and to bring fat in and burn your fat. And so that's wonderful in terms of people get a lot of weight loss in a very short period of time. The problem is what you've done to your metabolism is indicated through the way that you're eating is indicating starvation because you're not bringing in the carbohydrates. Unfortunately, your body has a mechanism in terms of what it sets your fat stores. And so when the body encounters a starvation event, how it interprets a starvation event, what happens is as soon as you stop that diet and you start increasing or having any carbohydrates, the body says, oh my goodness, I'm going to starve later, so I am going to take in as much as I can and I'm going to convert it to fat. And where your body weight was, it's going to increase that set weight to where a homeostasis now is more weight. And so when people start doing very extreme diets, they tend to lose weight, feel good, and then go back to normal. And their normal then sets their weight higher. And then they do again, and they do again, and they do again, and they do again. And you're setting yourself up to always have a higher set weight after that particular type of diet.
So it would be beneficial to slowly reintroduce carbs after giving keto.
If you've been under keto for a very long period, as soon as you start introducing them, the body is going to snap back. You can't retrain your metabolism because you've encountered what it interprets as a starvation condition for a very long period of time. And so what you do is you don't have that long period of time so that you don't reset your metabolism to lose weight. Well, it went on slowly. It has to go off slowly. You keep your biochemistry and metabolism going the same way. You just increase the amount of burning versus what you take in, and you do it slow incrementally so that you don't shift your metabolism in any way. The good thing about these diets is that you lose weight very quickly. And that's what most people want to do. They want a quick fix. But unfortunately, a lot of people set themselves up in a cycle that will continue their weight gain over their lifetime.
So what do they do with diabetics? Do they have medications?
Yes. Depending upon which diabetic condition that you have, if you aren't producing insulin, you take insulin. And or you take other medications that allow insulin to work better if you are producing a little bit. If you are of the type where your insulin is high and your receptors aren't working, then adding insulin doesn't help. What you have to do is a diet. You have to really do it by diet. And so the fact if you are insulin dependent, you can get to a place where you actually have a pump installed, and so you don't have to take shots all the time. There are ideas about those pumps actually having sensors for glucose so that you don't take your own blood all the time and watch it. The unit could both sense your blood sugar and inject insulin at the proper amounts. That hasn't been FDA approved yet because if you inject too much insulin, you can put yourself in a diabetic coma. If it doesn't sense too high, you can put yourself in a diabetic coma. And so having a manual check right now is protective as long as you check. So some people don't check and some people don't use the insulin that they need to. But most people do this. If you're going to go into the doctor, you're diabetic and you haven't been compliant, you are compliant the few days right before you go in, your blood sugar is perfect. That's why they go check what's called the A1C. The hemoglobin reacts with the sugar, but only when it's high. And so your A1C is an indicator over long periods of time where your blood sugar concentration has been. If it's normal, you've got a numerical value that it falls between a range. But if you go in, you have perfect blood right now, but for the past 4 months, it's been really high, your AC1 goes very, very high. And so you check both the blood glucose and the AC1 to find out where you're going. And so we can get, if you're under starvation, the molecule that's roaming around in the bloodstream that has your energy is these ketone bodies. Once you get out of the liver cells, the hepatic cells, that's where ketone bodies are synthesized. If you get in the muscle, this is broken down, and in a reverse where you'll have acetyl-CoA. In this case, the muscle is not sending the amino acids out as glucose. In this case, the muscle is sending all of its carbon chains into the citric acid cycle to make oxaloacetate, and now this acetyl-CoA can enter in the citric acid cycle and make ATP. And so in the liver, under starvation conditions, you have none of this, but in the muscle, you can fund this from amino acids, and that's why I can use the ketone bodies in the muscle, and I don't use them in the same kind of reactions. I don't use them in the liver where they're synthesized. And so acetyl-CoA is a huge kind of crossroads. If you think about all of metabolism being streets of a city, there are certain intersections that are really, really important. Acetyl-CoA is one of those intersections in which a lot of different pathways all come together. And so the control of how acetyl-CoA in its concentrations and where it gets sent is one of the key places in which metabolism is balanced and regulated, because it can go into the citric acid cycle, it can go into the ketone bodies, it can be converted to acetone in a bad reaction. I can make acetyl-CoA from fats, I can make acetyl-CoA from sugars, I can make acetyl-CoA from amino acids, and so there are a lot of ways I can make the acetyl-CoA. Acetyl-CoA can also do synthetic processes, which we haven't got to yet, but it will be a crossroads to synthesize cholesterol. And so this is of the structures we've seen around. Glucose has been a really big kind of interplay about where a lot of places it can go. Acetyl-CoA is one of those other big molecules that go in a lot of different places.